Hi all, I apologize for providing a pre-recorded talk, but this is supposed to be at about 3 in the morning local time where I am in Italy right now, and I was somewhat worried about my ability to be coherent or even wake up at that time, so this is probably better for everyone. Anyways, I'm going to tell you briefly about a new set of reporting guidelines and a checklist that we have produced, and try to convince you to start using it in your role as author and also as editors and peer reviewers. I want to just mention as background a project that motivated this study in the first place. This was an effort to create a database that I call BugSigDB, which is a standardized database of microbial signatures and information about the studies that produced them, such as what health conditions were under study design, alpha diversity measures, and differentially abundant taxa. This is one that my lab has been working on for a couple of years now, and it's in its final preparation stages. We've curated about over 500 microbiome papers into standardized formats, a couple of thousand signatures, and about 30 curators have worked on this. And in this process, we've had weekly review meetings of papers that revealed widespread heterogeneity in how microbiome results are reported. I won't say any more about that project, only to say that it is in beta release and you're very free to try it out. You can find published studies, the experiments or contrasts that were performed for analysis, signatures that resulted from them, and where any given taxa has been reported in the literature. One of the things that really stood out to me in reviewing all of these papers is how much work curation is and how much more work that it should be. These are just a few examples that I've collected of different ways in which the main finding of these papers, the differentially abundant microbes, have been recorded, and the kinds of formats that they have to be manually extracted from to produce a list of which are the differentially abundant microbes. So just to say, it takes a lot of reading and very close studying just to figure out which of all of these different, in this case, genera or species in these different formats were actually different between the conditions of study. So we set out to create a checklist that would make this easier for the people publishing studies to put all of this information, results, methods into a format that anyone could understand easily. And this is primarily the work of a doctoral student in my lab, Chloe Mirzai. So we set out to make a checklist based on existing checklists, but focused on microbiome studies that would be intuitive and straightforward for people to use across different fields of study, because it's a very multidisciplinary field, microbiome studies, that would not only provide a checklist, but would help authors to write well-organized microbiome papers, and that when used would provide easier comparison of study designs, methods, and results between papers. And as a general guiding principle, we aimed to be method, database, and analysis agnostic. So we did not aim to say which methods should or should not be used, only to ensure clear reporting of all of these things so that reviewers would be able to assess them easily. The reason behind this is that we're far from consensus on a lot of aspects of microbiome data analysis methods and laboratory methods, and these ideas are changing, and they do depend somewhat on the type of study being done. So we did not want to get into that. We just wanted to make it clear for anyone to assess what was done and what was found in a study. So how did we go about developing this checklist? This is the basic steps that we followed. A few of us, a core team, primarily Chloe Mirzai, Professor Heidi Jones, a faculty in epidemiology in my department, and myself, drafted an initial checklist, and we drew upon where we could the existing checklists in the field of epidemiology, and asked a few other people such as people from the Genomic Standards Consortium who have experience with checklists, and came up with an initial outline. And from that initial checklist, we emailed it widely to the microbiome community to invite collaborators and participation in checklist development. From that, we collected feedback via email and a Google document, and then synthesized that as a response to reviewers. So this turned into a letter, like as if this were a peer review process. And then we repeated these steps 2 to 4 until we had no further comments, which resulted in about a 50-page single-spaced response to consortium feedback letter. And we included that response to the journal, hoping that it would encourage reviewers there not to try to overrule the consensus of about 100 diverse people from the microbiome field. This consortium is now over 100 members from about 70 institutions, very multidisciplinary, people from all areas: microbiologists, bioinformaticians, epidemiologists, biostatisticians, and physicians. And I do want to encourage everyone here to come and join us. There's still plenty of work to be done, and this consortium will continue its work in improving the reporting of microbiome studies in the future and assessing our efforts to date in doing that. If you do want to join, you can contact me directly, or you can go to our website, stormsmicrobiome.org. You can also join our mailing list. Just briefly about the checklist itself, it has 17 items containing 57 sub-items divided into sections that correspond to a paper outline. And this was an intentional decision to make it both to make it easy to use and especially to encourage authors to use it at an earlier stage before they go to submit it to a journal to help structure the paper. And particularly for people who are either students or they're new to the microbiome field, or really anyone who just doesn't have experience across all the areas of a microbiome study. If you are a microbiologist, then the epidemiology components of it can be helpful to you. If you're an epidemiologist, then these laboratory components, et cetera, can be useful for you. But it looks something like this. There are these items with recommendations. There's additional guidance, which is not shown here because it's too much to show. And then a yes, no, NA field, and a field to enter comments where you can actually answer these things here in the checklist if you want to. That makes them very easy to find from the checklist, or just point to a location in the document or explain why you've answered no or not applicable to this item. Also accompanying the checklist, and this is a figure in the paper, I want to call out this very nice analytical sample size flowchart, which is credit of one of the organizers of this workshop, Francine Marquez, who developed these super useful flowcharts that really are applicable not only to microbiome studies, but to any observational or experimental study that involves the recruitment of subjects and some dropout along the way. So you're free to use this. I highly recommend using it in any such relevant study. The checklist is now published. It was published in Nature Medicine just this last month and is now required by most of the Nature family journals for any article relating to microbiome studies. And we're now working to encourage its use by other authors and journals. I think it has a number of reasons that you would want to do that. It facilitates peer review, makes the work of peer reviewers more efficient and easy. It improves the quality of reporting and the replicability of studies, and it can also help readers in finding key information, at least when the comments field is used to enter information or to point to the particular point in the paper where something is discussed. In some next steps, we're now working to assess inter-rater reliability of the checklist when used as an instrument. And we're looking for volunteers for this now. That would involve reviewing a few studies using the checklist, and then we would analyze that by comparing the responses we have when multiple readers apply the same checklist to the same paper. And we're also looking to develop a machine-readable version of the checklist. So please do get in touch or join the consortium if you're interested in taking part in any of this. Thank you to everyone who was involved. A number of people from my lab were involved in the development of the checklist and all of the work that went into the BugSigDB project during it. The many student curators who were part of trying to read and understand and discuss published microbiome papers, and to my collaborators on this project, including Francine Marquez. Thanks everyone very much.